Good morning, and thank you for joining us for this panel discussion on rare and ultra-rare diseases. I'm Susan Slagenhop. I'm the Scientific Director of the Mass General Research Institute and an investigator in the Center for Genomic Medicine at Mass General. Certainly one of the most innovative segments in all of healthcare is the development of gene and cell therapy, gene and GCT-driven therapies for rare and ultra-rare diseases. And we've seen some successes. Often these advances are unique in that they're driven in part by a combination of disease foundations, philanthropists, and venture funding. The goal of making these breakthroughs routine and affordable is challenged by multiple issues that I hope to discuss today with our panelists. Joining me today is Leah Bloom, SVP from Novartis Gene Therapies, Bobby Gaspar, CEO of Orchard Therapeutics, Emil Kakas, CEO of Ultragenyx, and Stu Peltz, CEO of PTC Therapeutics. So thank you all for joining us today. And let's get this conversation started by talking about what is driving the interest in rare disease and what are the key barriers to breakthroughs. So Leah, maybe I'll start with you.
Sure, thank you very much. And thank you for having me as a part of this panel. So first of all, I would just say that rare diseases, as we know, are typically monogenic in nature, and their very clear etiology and straightforward biology really therefore make them ideal targets for these genetic medicine approaches. And I think there's really three things then that are driving the interest in rare diseases. The first is, as we mentioned, it's the paradigm shift in technology innovation, fundamentally built on the foundation of the sequencing of the human genome and then following by dramatic advances in genetic medicine, including gene therapy. The second is the tremendous impact that's been demonstrated by early treatments that have become available to patients in the clinic and commercially. So for example, this would include Luxturna for retinal dystrophy and Zolgensma for SMA. And finally, it's the recognition of this value in the health systems that really enables sustained investment in these rare diseases. These developments also have been obviously well recognized, with around 20 billion in funding flowing into cell and gene therapies last year. Obviously, many of these are for rare diseases. What I would say is challenging, however, is that a lot of the larger and more tractable diseases are getting a lot of attention, leaving some of the smaller diseases underserved. And this is further exacerbated by just the limitations to the market opportunity in developing these one-time versus chronic treatments.
Mm-hmm. Thanks, Leah. Emil?
Well, I think Leah put forth a lot of the important things. I think the technology and ability to go after cells, not just gene therapy, but all the different strategies have really changed things. And with a genetically defined disease like this, I think it really opens up the door to having a very high success rate and actually having a very important and powerful effect on patients. I think one of the key challenges, though, that was going to happen is that these treatments are expensive development, and for some of the small ones, the ultra rares, we'll end up developing these treatments, but will the system allow access globally? And I think that's where we were running into quite a few issues. The challenge we also see is that the cost of development is going higher and higher, where more and more demand is being placed on how you do development, all the extra things you have to do for development. And as that burden rises, then the cost or the recovery of the system is going to get worse. I do think we have to get better at qualifying biomarkers to approve off smaller studies and get drugs approved more quickly. And that will help open the door to managing costs of things. But if the development demand is higher and higher for rare diseases, as it has been, the ability to develop treatments for ultra rares gets even worse. And I do worry that we won't be able to take advantage of the great science we have if we haven't figured out the system with regard to getting things approved promptly, efficiently, and to take advantage of that technology. So those are some of the things I'm thinking about when I look at rare. It's exciting, but it's also daunting at the same time.
Exactly. Stu, I'll go to you.
Yeah, I think what you've heard, I think, covers most of it. From our perspective of, we've been for the last, quarter century working on monogenetic disorders of ultra rare nature. So the issues sort of remain the same, and we've seen some of the successes because of some of the choices where you can actually see improvements from where a patient was before. Mm-hmm. And that makes development and ability to approve those easier than for instance, say neurodegenerative or neuromuscular diseases where there's a continuum where it goes down and patients may not necessarily improve, but slowing or stifling the further is key. But it's hard, I think, as Emil said, is that, we're dealing usually with imperfect natural history, imperfect outcomes, small patient populations. And when you think about the monogenetic disorders and the number of diseases which might be construed as commercially viable, they're much smaller than you might think. And as such, it makes it, so if you have a particular disorder you're trying to treat and has 500 patients, then as Emil said, if you're required to do all the clinical work, there may not even be enough to do that. And then certainly they're all treated by then, and then there's no commercial venue. So people are going to have a more difficult time choosing those. So I think the notion is, how do you do something like this with gene therapy and be able to have approvals and post-marketing registrations or things to be able to, especially for the smaller ones, to be able to demonstrate activity and approval in the long run. That's, I think there's going to be some real challenges there.
Yeah, agree. And Bobby, your thoughts on breakthrough and what's driving interest and key barriers?
Yeah, I mean, I think as far as what's driving the interest, I think the panelists have really commented on, in many of those, the high level of unmet need in some of these, ultra rare conditions where there's often no or limited or unacceptable current treatment options. The monogenic nature, which gives a certain tractability, one gene is missing, replace or add that gene, and then you can get a therapeutic effect. The kind of gene therapy that we've been doing, which is hematopoietic stem cell gene therapy, where you're taking autologous hematopoietic stem cells, introducing a working copy of the gene, and then returning them. Because of the self-renewal nature of hematopoietic stem cells, it does mean that a single administration of the gene-modified cells can have a long-lasting impact. And if you take the analogy with bone marrow transplantation, it's essentially a lifelong effect, so it is potentially curative. Now that has huge advantages, and we've already seen that in the in our clinical trials and in the approvals that we've seen for some really very severe and devastating diseases. But also, as you can imagine, if you're just giving it one time, you can have other challenges as well in terms of reimbursement and access. And that's a paradigm shift for the system that needs to be, that we all need to deal with, because we see how important it is for patients. But then, how does that reconcile with an economic and reimbursement model? So that's one aspect of it. I think the other part that may have been touched upon is the fact that the use of HSC gene therapy or gene therapies in some ultra rare conditions also kind of gives clues to what might be applicable in larger indications as well. And so the study, and this is both in genetics and also in therapeutics, but the study of these ultra rare conditions, they often act as models for what might be possible in larger diseases as well. And I just take one example. We've treated a condition called metachromatic leukodystrophy. It's a very severe neurodegenerative condition where the ability to use hematopoietic stem cells to cross the blood-brain barrier and deliver therapeutic genes to the CNS has been able to demonstrate the stability of neurocognitive, of neurodegeneration. So we've been able to prevent neurodegeneration. Now, that kind of gives you all kinds of clues about, how hematopoietic stem cells can get into the CNS and deliver therapeutic genes. And if you can do that for an ultra-rare condition, can you use that for other larger forms of neurodegeneration? So it's just one exemplar of how you can look at the use of these conditions for larger diseases. I think just on a personal note, I started off as a junior doctor looking after patients with a very severe immune disorder, never thinking that this would end up leading to the formation of a company and everything else like that. But it really was because of the impact, the clinical impact that you see of this technology in these diseases that kind of drives you forward to thinking, well, that just opens up the door for not just this condition, but for many other conditions as well.
Exactly. That's certainly the goal. Let's talk for a minute about the role of natural history studies. For many of these diseases that we're trying to tackle, the natural history is not well understood. The diseases are extremely rare, as we know, and extreme variability patient to patient. So let's talk for a minute about the role of natural history studies as we move forward. How important are they? And how do we justify the cost? These things are very, very expensive. So how do we justify the cost, and are they needed in every case? Whoever wants to jump in.
Well, I'll jump in. I'll just say, natural history can be useful and fruitful, right? Because it helps you define where the changes are. I mean, ultimately, when you want to get a drug approved, right, you want to know what's the fastest way to demonstrate a change,
Mm-hmm.
and what's the best patient population to demonstrate that change, right? So if there's good natural history, at least you get some ideas on that. I will point out that natural history sometimes does differ than, say, a placebo control, just because of the nature of but at least, you need to start somewhere.
Mm-hmm.
So, I mean, from our point of view, at least it's valuable information to be had to try and pick out the fastest way to get it approved. So, I mean, we've always tried to do some natural history to try and understand the best way to get it approved. Now, there are some downsides, and it is expensive and it takes a long time, but that's where I think also patient advocacy groups and others can be helpful, where, early before there are treatments, if there are natural history that people do that could be useful, I'd argue that that's one area where people can contribute.
Right.
I agree with what Stuart was saying. I do think the one challenge with when patients do those studies is, have they developed the right endpoints that they're actually measuring? Because very often they're using things that are laying around that people have been doing and may not necessarily. Now, some foundations have done a good job of developing endpoints, but if you measure things, scores of various kinds that are not the right thing, creating a lot of natural history data on that may not necessarily take you anywhere. I do think natural history is important, but I do think it's a little bit overstated. The truth is, for a lot of our programs that we work on, if the disease is going to take 5-10 years to develop, doing a natural history to demonstrate things is basically non-viable for the company to do. You can't spend 5 or 10 years waiting to get answers to things. For programs where the disease goes down is very fast, for example, infantile Pompe or late infantile Batten's disease, where you have a 1 to 2 year time frame, the natural history that can really define if there's a stereotypical decline rate and very well described, and that information can really helpful. In those cases were used as control information in a trial. But a lot of people, a lot of patients believe these natural history can be used as controls. It's actually very hard to do if it's not a very rapid decline kind of effect, a very major rapid decline type effect. So the natural history is rarely going to get rid of a controlled study design. And that's one thing that people get missed. The second thing is people say if they have the natural history and know the variation, that they'll know how to design the studies. Well, you'll know something about it, but it doesn't solve the problem if the patients are heterogeneous.
Right.
The fact they're heterogeneous means they're heterogeneous, meaning when you start enrolling them, they'll be heterogeneous again. They'll have various things. You could try to select your slice, but that slice may not represent all the patients. So often we learn they're heterogeneous, which is basically we knew that already, but you can try to get a little more information. What I actually suggest to people is before you run off and do a natural history study is do a large single measurement cross-sectional study of your population. It's a lot more better to spend your money getting 100 patients screened on one day just to understand the breadth in a cross-sectional study, learn, develop your endpoints a little bit, and then think about a repeat dose natural history. It's a far smarter way to go. You'll get a lot more information. You'll understand heterogeneity, and you can use the history of that patient to develop some information about how fast they declined. And so it's a lot faster and actually ends up becoming more effective. For a disease that goes slow, natural history can take a long time. And in the end, it's really challenging. It's a really challenging thing. And I will tell you, all the products I've been involved with have gotten approved did not have a full-born natural history study involved. Right. And so that's not to say it shouldn't be done. I agree with Stu that patient groups should be focusing on that to get some of that long-term data. They should do some work on the endpoints too before they go diving into it. And truthfully, very few of them have the kind of money involved to give you a regulatory appropriate quality data collection system, right? The Excel spreadsheet taken to the FDA doesn't work for a data collection tool. And we've had a number of patient groups go to the FDA and say, here's our great natural history study. The FDA says it's interesting, useful, but you're not going to use it as a control, and you're not going to really submit it. You're going to use it just to help inform the professionally done work. So a lot to do there with natural history. I think it's important to learn about disease, but I also think there's ways to learn that might be more efficient for disease that take a long time and for which you can't spend 5 to 10 years watching them.
Right. Good thoughts. Leah or Bobby?
Yeah, I mean, maybe just to put a finer point on some of those, and I largely agree with the points that have been made. I mean, from a type 1 SMA perspective, at least from a trial design point of view, we were quite fortunate to be able to move quickly. When we and many of the other innovators have taken a similar approach. So we recently did an informal poll of around 70 clinical trials in our space, and the vast majority of them are uncontrolled. Now, a lot of those obviously are phase 1 trials. But still, while there's a lot of expressed interest in natural history, we do see from the FDA that there's a hesitance to replace these control arms with natural history for some of the reasons that have been stated, especially for these more slowly progressing diseases. And on the ultra rare diseases, I would say, not only can it be challenging from a cost perspective to make this work, but just from a pure feasibility point of view, if you only have 5 new patients every year, actually, finding those patients and then tracking them for a long enough period of time for that data to become useful is extremely difficult. And so I agree with Emil that we need to get more creative, continue to equip advocacy groups with tools, make better use of chart reviews, think about where we can augment, if not replace, with natural history data that's been well established.
Thanks, Leah.
I won't kind of repeat everything, but yes to the kind of the challenges, yes in such small populations. I think in terms of the understanding, from a kind of almost academic learning perspective, to understand where the challenges are for the disease population, I think it's incredibly important. In the regulatory setting, in terms of comparing it against your treatment, that's where I think a lot of work has to be, because actually in some diseases there is no other alternative, and and the regulators want to compare against something, and all you can compare against is the natural history. And so the question then is, how do you construct that natural history? You can't do it prospectively, because if the natural history is death within the next couple of years, how could you randomize to that, or how could you do that concurrently? So you then have to go back to some kind of retrospective natural history collection. And I think that's a really, really important point to make to regulators, that there is no alternative. You have to look to natural history, and maybe we could talk about what kind of natural history and what kind of level of scrutiny of that natural history collection needs to be put in place. But especially when there are such small numbers of patients, especially in these ultra rare indications, you might have had to have done that over a decade or so to collect that kind of data. And would you do it again just in order to have your comparator arm for approval, or is there some way of making use of what you already have, however imperfect that might be? So I just think there are some practicalities. If we really want to make medicines for these ultra rare conditions, and we can have this quantum of benefit like we've seen in SMA and other diseases, then let's think about how best we can do that.
Right. Your comments are a perfect segue, because where I want to go next is to the regulatory side, and thinking about how we keep moving forward. How do we change clinical trial design, or use biomarkers? And I believe Emil mentioned the use of biomarkers, to measure target engagement, other things that are out there. How do we keep moving forward on this?
Well, I think it's been particularly challenging on the biomarker front, because I think both the regulators as well as the reimbursers have kind of basically disparaged any biomarkers meaning anything at all, even ones that have been validated. Right. And it's a tone that's basically about restricting the spend for drugs than it has anything to do with science, right? The truth is, and it's a major mistake, that we believe that the clinical endpoint is more meaningful than the biomarker endpoints. The truth is, the biomarkers can be far more accurate measures of disease than anything clinical. And the example I'll give you is the whole HIV story. Do you think we'd have gotten to highly active retroviral therapy, multiple drug-based therapy, using anything clinical at all? We would have never gotten there. We would have never achieved that. The truth is, the viral load, CD4 counts, those were the ways we get to treat that disease that takes years to progress clinically and is highly variable. The same is true in rare diseases. They're highly variable clinical because of secondary genetic factors. But the biomarker will tell you what's really going on in disease activity. A biomarker that's proximal near the beginning of the disease state. And that is what we need to get better at. That's advancing the science toward getting it right and doing it up front. And we need to get better at that and stop looking at it as a compromise and looking at it as an accurate forward way of assessing true disease activity. And I just think that we're quite mistaken. In the MPS world where I've been, for example, urinary substrate, we've shown highly predicts clinical disease. It has correlates across all the tissues. It's highly predictive of what's going on inside tissues. And it'd be a far better way to develop a new version of treatment for MPS 1, let's say. There's enzyme therapy. But if they keep requiring clinical endpoints, which are highly variable, which require very large studies, and the studies, the follow-up improvements don't get done. So we have enzyme therapy, but they can't get the next version. We can't get a better version. They have to do a walk test again. They have to do some other things, which require 50, 100 patient, naive patient studies, which they can't get. And trying to look at good to better is an even smaller distinction, right, than good to placebo, right? So well, we basically shot ourselves in the foot. We're failing to actually accelerate our optimization of treatment by not using biomarkers, which are a more accurate representation of disease. So I'm giving you the stronger view of this, which is it's not just cheap and easy, it's actually smarter and better.
Yeah.
And we need to get our heads around that and start doing the right thing. And we would actually accelerate drugs, reduce the cost, and get more patients treated. And I've written a whole paper on that, how using a biomarker program would reduce costs of development. I did it, modeled it for 15 diseases, about a 70% reduction in the cost of development. And think of then what you could do on pricing and access if you had that much reduction in time and development. How many more diseases we would treat, rare and ultra rare. It would change everything. That's why I fought so hard for that. But the world is going against it right now, and I need to turn the tide because it's totally wrong.
Right. Well, I see other people's heads shaking. I think there's agreement on the panel. I'd like to hear from others. I couldn't agree with you more. I think there's many instances in these rare diseases where we can measure, using a biomarker exactly what we're trying to target, and yet the FDA won't listen to that. So please, Leah, do you have thoughts to share with us on this? I'm sure you do too, Stu. I'll get to you in 1 second.
Yeah, no, absolutely. I mean, agree that the thoughtful and creative application of the biomarkers is critical. I was actually going to take it in a slightly different direction and also talk about the further development and characterization of different platforms, and then the application of our understanding of the platform properties to be able to develop treatments more efficiently for new diseases. This is obviously something that's gotten a lot of attention from Peter Marks and others, and where there's a lot of effort from an AAV perspective. And I personally like this notion of pursuing this extreme examples to figure out how far we can take it. I haven't found a good analogy, but maybe think about like a Ryanair disrupting the airline industry, where we can work collaboratively with FDA, for example, to advance treatments for ultra rare diseases and of less than 100. Think about where we can leverage known platform properties to make this a more efficient proposition. And I think if we do that effectively.
Whoops.
Uh-oh.
Okay, that's okay. We're going to go straight to Stu while Leah comes back.
Okay.
Okay, let's see how long I last. No, I mean, the notion of biomarkers, obviously, we all like, but we all know that the FDA is going the other way on that most of the time. And you can look at like dystrophin and DMD at the point almost broke the FDA apart as a consequence of that. And so I think they'll take like natural histories and others when it's pretty clear, like developmentally arrested. A good example is like the SMA, right? Where you know they're not going to get their type 1. It's pretty easy to see if they never would raise their head or roll over or sit or stand. They'll accept that natural history, but they won't. I don't think they're very, while there's always talk about real world data and examples of this, it's hard for them to want to accept that all the time and compare it to it. It sort of goes against their gestalt and the nature of how the FDA is built. At least that's the way I see it. So it's I'm all for, especially for biomarkers, especially in ultra orphan diseases where there's just not the people. I mean, we've worked together on things where it's just not almost doable to do a real clinical trial.
Right.
It's doable probably to do to demonstrate some activity, but not, and then have a post-marketing commitment to follow them. But to be able to do a real clinical trial, it would be absurd. And so at the end of the day, you're not going to put, I mean, the discovery, development, commercial muscle behind it if we don't figure out a better way to make it doable.
Right. And we don't have much time left. But I did want to, and Bobby, I'll start with you, ask each of you how closely your companies work with family organizations and the role of these patient advocacy groups in moving us forward on the regulatory front.
I mean, I think it's essential. It's absolutely essential. I mean, these are such rare diseases where there isn't a huge amount of literature out there, and you can only say so much with graphs and tables and data. And when you hear a patient speak about what they have, or a family speak about what they have been through with their child and what other families have been through, just the kind of the emotional trauma and the just the suffering that the families go through, it speaks so much more than all of that. And sometimes I think the regulators just need to hear that, just to hear what the effect upon the families are and what a difference a treatment can make. And I think that just kind of changes the whole sentiment about what can be achieved. And so I think it's hugely important.
Thank you. And we are out of time. So I want to thank each of the panelists for this discussion. Certainly we could, I'm sure, discuss this topic for another couple hours, but I really want to thank you for being with us here today.